The last biochemistry reaction that we're going to talk about is my favorite. It is nucleic acids, which is a global term, and when we usually talk about them, we're talking about two specific type of nucleic acids. There are a few others that have now popped up, like siRNA and things, but the two that we're going to really focus in on are DNA and RNA. DNA stands for deoxyribose nucleic acid, and RNA stands for ribose nucleic acid. A slightly different sugar makeup, and it gives them a little different properties, which we'll talk about in a second. Their function, they really are the instruction manuals that make us who we are, and they're basically instructions on how to make those proteins. So when we need to make those proteins that we just talked about, we go to our DNA, and our DNA will send us a temporary message called an RNA, a temporary sort of message, and give us the instructions on how to make that protein. And when we don't need it anymore, the instructions sort of disappear, the protein can get recycled, and we can make something else. So that nucleotide is made up of 3 parts. There's a 5-carbon sugar, so this would be the sugar. There's a nitrogenous base. Those are those A's and T's and C's and G's you might have heard about in DNA. And then finally there's a phosphate group. And a phosphate we're going to talk about more soon, but they basically give it energy, and we can do different things with different phosphate groups, but we'll talk more about that in a couple weeks. So if we look at the nitrogenous bases, and I'm going to skip one for a little while, there are those A's and T's and C's and G's. You'll notice that there's the cytosine and thymine. Those are both in DNA. There's adenine and guanine. And these guys have a double ring. They're called purines. And these guys, all 3 of these guys have only one ring, and they're called pyrimidines. So this is those C, that T, and the A and the G that you hear about with DNA. And then with RNA, the only difference is that this thymine, we don't have it. We basically replace it with this guy called uracil, and that's why uracil, it says it's in RNA, and this one says thymine is in DNA. That's basically one of the bigger differences. So DNA has 4 bases, those A's and T's and C's and G's. And you remember I just told you adenine is what the A stands for, thymine stands for the T, cytosine with the C, and guanine is the G. What you're looking at over here is these things called hydrogen bonds, and we talked about it with chemistry, and I said that, you know, it's one of the weaker bonds, but it's really important when we come to these kind of base pairing rules. Well, it turns out that when we look at these A's and T's and C's and G's, between A and T, I think there's a little typo here, so A and T has 2 hydrogen bonds, and G and C has 3 hydrogen bonds. So these should be reversed. It should be guanine and cytosine have 3 hydrogen bonds, and adenine and thymine have 2 hydrogen bonds. Why this is so important is because you can see, like for instance here, A and T, they have 2 hydrogen bonds, and G and C should have 3 hydrogen bonds. So it gives that DNA its strength of holding together, but if you recall back from our chemistry chapter, A and T, hydrogen bonds are actually one of the weaker bonds. So because they're so weak, we can actually open and close this DNA like a zipper. We can separate them out and get access to these A's and T's and C's and G's, which their combinations tell us which amino acid to use, which we'll talk about in a couple weeks. And then when we're done, we just let go of the DNA, and it actually auto sort of comes back together because of these hydrogen bonds to stay together. We'll talk more about that in a couple weeks. RNA has 4 bases. They have the adenine, cytosine, guanine, but it doesn't have thymine. It has this thing called uracil. And uracil gives it sort of it's a different nitrogenous base, and it also makes it a little more temporary. It comes apart after 20 minutes. The differences between DNA and RNA are that DNA has a deoxyribose nucleic acid, where RNA is called ribose nucleic acid. DNA has a different sugar, and that's really the deoxyribose sugar, and RNA has the ribose sugar. The bases that we kind of talked about already, DNA has an A and a C and a G and a T. RNA has an A and a C and a G and a U, so the U and the T are the difference. But the biggest difference is that DNA is double-stranded and RNA is single-stranded. Also, RNA is temporary. RNA lasts about 20 minutes in the body, where DNA lasts for, kept under the right conditions, could last for decades. So this is a lot longer code. This hangs out a lot more, and some of it has to do with the double stranding and the hydrogen bonds, where this guy doesn't have that extra strand, so it makes it weak. The other thing that makes it weak is this uracil instead of the thymine. We'll talk way more about DNA in the next couple of weeks. And then I've gone ahead and made this review sheet where it basically talks about each of the different macromolecules that we discussed, the monomers, the smallest component of each, the bonds that hold them together, their function. This is a really short list. This could have gone on, and the examples. Just a few examples of each. And granted, with proteins and lipids and sugars, these lists could have gone on for pages, but these are just a couple. Great. Well, good luck.